Так, ну что, у нас, наверное, рекомендации начать заседание. Так, дорогие коллеги, значит, меня проинформировали, что... Сейчас, алло. Да, значит, меня проинформировали, что все докладчики у нас, так сказать...
Thank you very much. I have been informed that we have all the speakers. Several of them are here offline, and 2 will be joining us online. And we are going to speak about vaccines, race for the evolution of SARS-CoV-2. So, So I will be moderating, and my co-moderator is not here with us, so I'm not quite sure how we are going to work. Maybe he is going to join us online. I'm giving the floor to the first speaker, Ivan Suslaparov, from the Research Institute of Virology and Biotechnology, Vector. And he's going to speak about peptide vaccine Aurora COV. From development to introduction. The floor is yours. Thank you very much. I'm going to begin. When 2 years ago the pandemic started, the Vector Center immediately began these developments, and at the beginning of 2020 we were working on vaccines. And after analyzing different variants and different platforms, and those were inactive vaccine, and then virus similar elements, then RNA or DNA, live and inactive, so on and so forth. And after analyzing all those platforms, a decision was taken to use alternative approaches in order to accelerate this process. At that time, we didn't have much information about the structure of proteins. Actually, nothing was known at that time. No epitopes were known, and the tertiary structure of this protein was not quite clear. And we decided not to use this subvirion vaccines. And there are so many various immunosuppressive domains, and there are non-immunodominant epitopes. We wanted to choose some most conservative section to avoid all these mutations in the future. And that is why synthetic peptides became a major platform, because this ensures elimination of the above-mentioned problems. So you see here different stages of development. First, design of these epitopes and peptides. Then we selected about 15 interesting peptides. Later, they were conjugated on a certain protein carrier, and then there were experiments on animals. And then we selected 3 most promising peptides. This composition was approved, and 3 lines were received, and then we moved on to preclinical and then clinical trials. What are the advantages of these synthetic peptides? It is very quick design, so it requires days from the trial version production, so 1 month maximum. Then biosafety of this synthetic peptide vaccine, because there's no pathogens in it, no biological contamination, then no virulence or some other reactive agents. And biosafety is quite important. So at the level of design, we could choose those epitopes that did not lead to cytokine storm, and those that were similar to human proteins, because autoimmune response might be activated. And receptors for... So there was a danger to get something that might lead to this angiotensin binding. We were using information about tertiary structure. As I said previously, we selected 3 peptides. One of them. So this is the tertiary. Okay, one peptide is localized in immunodominant RBD domain, and 2 outside this. Okay, so in the first part of protein on the surface of a virus, and it is on the spikes and the pedicle. So, and this could block penetration or binding of this virus with the cell membrane. Here you can see 3 peptides, and you also can see the major difference of Epivac Corona vaccine and Epivac Corona N, which is called Aurora CoV. The major difference is that these 2 peptides that imitate the spike protein, they are combined in their homological structure, and the peptide which imitates RBD domain epitope is unchanged. So then the first variant of the vaccine was developed, and we used 3 groups. These were ferrets, hamsters, and lower primates. And we know that hamsters can have coronavirus infection, and there's certain level of fatality, sometimes 100%. Then ferrets, they also may have this infection, and their response is very good. There are some clinical signs. All this was fixated. And as for primates, we used macaque rhesus and green monkeys. Okay, they are not so susceptible to this infection, but it was also possible to analyze how they responded to the disease. Right, and these are the results of the study. There is results on hamster and ferrets, 14th day after the second dose. So 2 different doses of this vaccine were used. And after this second inoculation, it was shown that 100% had immune response. And if we increase the dose by 3 times, then it goes down. And if the antigen of coronavirus is injected, then the response is 2 or 3 times lower. After 2 inoculations, 94% develop specific antibodies. It is on the 14th day, and after the first inoculation. And after the second vaccination, 100% of ferrets demonstrate a stable level of antibodies. Immunoprotective qualities of the vaccine were studied in neutralization after intranasal infection. And again, it was on hamsters and ferrets. And here the results are on the 6th day. The body mass index was quite different. And in the control group that received placebo, it was quite different from the other group. The histological lung tissue analysis demonstrated severe pathological damages. And animals were vaccinated with a high dosage. They demonstrated only localized damaged spots. And for ferrets, on the 6th day from getting this infection, so the burden of viral RNA was reduced drastically. And this was a great difference between the placebo group and the control group. And the time of elimination among vaccinated animals is reduced significantly. Here we can see histological and X-ray analysis of vaccinated and placebo hamsters. As I said, see the vaccinated animals, they demonstrate some minimal damaged lung tissues. Then the clinical assessment of the severity of lung damage. We use several criteria. The hemorrhaging damage and others, and there's a score for each animal. And so we analyzed these focal points, then the level of damage, and histograms were developed, and it was obvious that the vaccinated animals were less damaged, or their lungs were less damaged than those animals that hadn't been vaccinated. Also, we used primates, and all these animals were protected from these lung damage. At the same time, in the placebo group, after infecting them with coronavirus, about 67%, so they demonstrated some signs of viral pneumonia. And So there were several different species of primates, and we registered some differences of temperature, body temperature, 1 degree Celsius in comparison with the vaccinated. And the body mass didn't change in both groups. And on the 4th, 6th, 7th, and 12th day, there wasn't any significant viral load in all samples, as it was not registered. And as for the speed of elimination, in some monkeys, it was longer than in other groups. And there are two major signs of lung damage. It is diffused alveolar damage and the hemorrhage syndrome. And this was observed in both groups, but in the placebo group, there were more of them. So the area of severe lung damage in the placebo group was twice as high as in the control group. During the preclinical trials, we were analyzing how prevalent viruses were changing. First it was alpha, then beta, then gamma, delta. Now we have Omicron. And we were analyzing how regions of our peptides are affected by mutations. And without knowing how this virus would be changing, actually we found the right spot. And this peptide that imitates these receptor binding areas, it is not affected by any mutations in different strains. However, the latest mutation in Omicron, it is on C end of this peptide, and it is 447 position. And our mathematicians and modeling shows that it shouldn't change the response. Recently, Chinese researchers published an article about universal monoclonal antibody neutralizing practically all COVID strains, and they have mapped this epitope, and our peptide is part of it. So this is another confirmation that this peptide is quite universal for different types of coronavirus, and probably it will be functioning for Omicron as well. So how long do you have? I have 3 more slides. How many minutes do you need? Because your time is up already. 2 minutes, please. Then I'm going to omit this slide. Okay, let's have a look at the clinical trial results. Here we show that for Epivac Corona and Epivac Corona N vaccines, Epivac Corona N works better. It provides the full protection, and here you see the results. The immunological efficacy after vaccination and the level of seropositive volunteers is 85%. This is for Epivac. And for Epivac N, it is 85.4%, so slightly higher. Also, it has been demonstrated that there are differences. So whether there are 2 or 3 inoculations. So 100%, it is on the 21st day. This slightly reduces 6 months later. Right here, these are the results of this research, and Epivac Corona N. We are completing the first and the second phases, and soon we're going to begin the third one. Okay, and here it is some information, okay, how this immunization is taking place and how we are evaluating the response. There's a special test system for checking efficacy of this vaccine, and the antigen in this system is the same as in the vaccine itself, right? So this is already manufacturing details. We have produced about 12 million doses, and now we are going to transition to Epivac Corona N. And recent independent studies have been published. And this is evaluation of effective parameters of different vaccines. So this is the table from this article. And they were considering safety, logistics, and so on. Logistics, that is whether it could be frozen or whether it is needed, and so on and so forth. So all those parameters were taken into account, and the score was calculated. And unexpectedly, Epivac Corona N is #1. So its positive advantages, its advantages were the highest. And here you see 15 different vaccines, and our Russian vaccines occupy the prize-awarding places. And what's next? We are planning modernization of this peptide vaccine depending on how the strain will be changing. Okay, so we are planning to modify. So you have used up 20 minutes. Sorry, we have to stop this session. So could you please finalize it? Right. We are planning to develop the endonasal form, and we are working on adjuvant methods. We don't have time for questions, maybe after this. Okay, the next report.
The next presentation is from the same institute by Ilya Simadinov, candidate vaccines against COVID-19 on the basis of recombinant strains of virus A and B. 15 minutes, please, and no more. Yeah. Good afternoon, dear colleagues. I'll be brief.
Меня зовут Имадинов Ильнас.
I'm Ilyas Simadinov. I'm the head of the laboratory of vector systems at this vector center. We, as one of the teams working in the center, is the task of developing the candidate vaccines against COVID-19. And my presentation is going to be in the format of the presentation dedicated to the recombinant strains, because the cultivation of the viruses and vaccines, virus preparations and vaccines is similar to what we do for flu. Allow me to start with the pathogen, beta coronavirus, with a genome about 30,000 ribonucleotides, and the share that is coding the structure proteins is 1/3. The spike about 12.5% RBD, that's less than 3%. Other components that are involved in the protective immune response coding a smaller part. Now, here in this slide, you can see the dynamics of the detection of the variants in the Russian Federation, and I will refer to that when I talk about the stages of development. And a few days after we presented the thesis to the conference, and the Omicron was detected, and in my presentation, I would like to speak about the stages of the work on the candidate vaccines against Omicron on the basis of this platform. So the first stage of the development of any vector vaccine is the design of the imagines and in particular the selection of the target antigens. In most of the vaccines, they are glycoproteins or receptors binding, and the nucleoprotein M is one of the components which is expressed in large numbers in the infected cell. The architecture of monogenes, in our case, immunogenes, it was in a few iterations native antigens and the signal peptide, autonomous polymerized options with the so-called system that makes it possible during the expression of this antigen to localize it. And similar other types of antigens in the presentation of the immune system. For the antigen to take longer time for the unloading from the system. Ilyas Remisevich, your slides are not being demonstrated. No, it's just the title page that we can see. We can see the first slide, the name of the presentation, but nothing else. Yes, no, this works. Yes, this works now. Right. So, what is this system of the reversal genetics for the flu viruses? It's a classical system suggested by Hoffman. It's 8 plasma system, 6 major proteins and two segments coding democratinine, which with the recombinant strains manifests the antigen properties. And this occurs experimentally. 8 plasmids are made in certain quantities, and interpolation is carried out at our center, the tested line for that purpose. And then MDCK, and then mRNA, synthesis of the protein machinery, replicative machinery, and then replication leads to the recombinant viruses and other saving of those or others. And we developed the technological platform for the reverse genetics and these viruses. And using these approaches, we work on 2 generations of the system. The prototypes, well known to everyone, and next in class, which is based on the live vaccines. Here you can see the main strains we're using in our work for the inactivated vaccines with respective mutations, and the well-known Russia SSR, the best for the live intranasal vaccines. So with the use of these particular variants, we carried out the development of the preparations. At the first stages, we used the strain Puerto Rico as a prototype, and the first goal we set was the place we were to find for the translation. We selected the segments 4th, 6th, or 8th, non-structural. And in the 4th segment, we were able to get the highest level of expression. But with further experiments with the concomitant infection, we were able to see dissertation, which actually excluded the possibility of the use of the 4th and 6th segments for the live candidate vaccines. For that reason, we mostly focused our attention on the 8th segment, coding non-structural proteins. And down below to the right, you can see the scheme. The 8th segment codes the proteins which are synthesized after expression, and the splicing is there also reflected. And then we can see the hybrid framework for reading, the processing that happens at the stage of the protein translation through to E2 peptide, and with the presence of the target antigen. The intercell transport occurs irrespective of the non-structural proteins. As I said, the first prototypes were recombinant virus on the basis of Puerto Rico, a very good strain with very good cultural properties. And at that particular moment, there were two versions, alpha and beta, which we were using. Briefly, the target antigen was modified in such a way that its end part was also associated with transmembrane, so it could be anchored on the cell and on the virions. And so this assumption was tested by us in the process of the purification with the ingredients. And so the results are displayed on the slide. The expression of antigens in the culture of cells was assessed with the use of the pyrophysical analysis of supermontant infected cells, and other methods represented here in the slide. These strains on the basis of Puerto Rico with the modification of the non-structural genome and proteins went down insignificantly, which makes it possible to assume that these strains are good enough for the production of vaccines. So with these promising results, we decided to move over to the live hyphenated adapted versions. And at that particular moment, we had the Delta strain in place. And so, and in this presentation, I would like to speak about two variants of antigens carrying transmembrane virion, and another variant with human antibody as one of the mechanisms with the presentation of in the immune system. You can see the main strains we were able to get within the altogether 4. And they code hemagglutinin RNA bus and prototype strains, which are recommended for the production of vaccines for Northern Hemisphere. We used these genetic structures, and the 8th segment was amended for the production of RBD. In this slide, you can see the results of the molecular mass assessment, and the molecular masses really coincide with the calculated masses, and these viruses with the cell culture were cultivated in a somewhat narrow way than the initial prototype variant. This is easy to explain in the context of the immune use of the recombinant variants. We were able to see that even earlier, they expressed, and so that's exactly what we were able to get. With the cultivation in MDCC, the titers were comparable, but these recombinant viruses were transmitted as well as the others for the experiments on the assessment of immunogenicity and protective properties. And the results will be ready by the end of this year. In the context of the application of this platform and this technology in the format of the current reaction, to Omicron, we would like to describe the advantages. First of all, the possibility of the joint work on the antigen components for flu and COVID, which is being used nowadays for the production of the vaccine preparations against flu, can be also used in a combined option for the production of vaccines against COVID-19. That's a quick way of getting recombinant strains, and particularly after Omicron emerged, we carried out the analysis, and we were able to get the substitutions that occurred in the genes. And within 2 days at the center, we carried out the seamless assembly of the full-size gene spike SARS-CoV-2. And important for the cloning of genes spike and RBD. And this will make it possible for us to examine this particular strain and whether it is dangerous or not that dangerous. At least we'll be able to analyze it. That's all I was planning to describe. Thank you for your attention. If there are questions, you're welcome with those. Thank you, Ilya Mesevich. And yes, no, you were exactly within the time allocated, so thank you very much for doing that. Any questions to the speaker, or we will have a later discussion? Now, since there is...
Okay, we see there's academician Zverev from the organizing committee. Okay, in the morning, there was time for questions. Okay, so there will be some time at the end. Right, now we are passing the floor to Maria Strukova and Dmitry Lyoznov. I'm not quite sure who is going to speak. They're from the Flu Research Institute in St. Petersburg. Okay, and they will be speaking about mucosal vaccines on the basis of flu A virus for prophylaxis of respiratory infections. Good afternoon, dear colleagues. First, I would like to thank the organizers of this conference for this opportunity to speak. For us, it is a great honor. And we would like to speak about mucosal vaccines, and probably it is the future, not the present, but the future, because this is the topic of this session. And we have been working on it for about 15 years, and we can demonstrate what are the current achievements and what we may expect in terms of vaccination campaigns. Right, this is Israel graph, and this is the country where a very large share, over 90%, have been inoculated, and this is 2 doses already, and many people received a 3rd booster inoculation. But still, we see these waves of the pandemic, and this is due to various mutated or new strains of the virus, and we can observe similar waves in other countries as well, where we see a large-scale vaccination campaign. We understand that the immunity due to vaccination, and currently we use injection vaccines, and we expect that antibodies will be developing in the body, and they play the main role in reducing the severity of the disease and the mortality level. But this way does not lead to prevention of replication of the virus in the respiratory airways. And because of this, the secondary infection, and there may be light cases, but this is passed on to those who are not infected, and we still see that the virus is spreading. In order to resolve these problems, we can use vaccines and various prophylactic systems. And this slide shows the situation in April 2021. And here, the countries we see here, okay, so they have expanded this work. This is on inhalated vaccines. Okay, it is United States, Iran, some other countries. Russia is not here, but we are working on it. We expect that in the future we are going to see these nasal versions. And this can be adeno, vector, or when we select certain adjuvants. Okay, so these are vaccines that are either inhaled, and for us intranasal, this is more common. Our experience of fighting the flu shows that when there's seasonality, when strains do not correspond to the vaccine, the only version that may protect is an active viral vaccine, because inactive vaccines may only be effective for the specific virus. Okay, so here we see it is information from 2007. We see that an active vaccine, live vaccine, was effective, but the inactive one wasn't so successful. Right, what do we know about this cross-protection? Well, these are T cells, memory cells, and this is the first line of protection, the first barrier on the way of the infection, and they respond immediately to pathogens. And there's induction of various cytokines, and it blocks the replication of the virus. And in such a way, it limits the development of this. And this is globulin A. And here we're speaking about the flu. And there's a separate population. It is not about cross-protection, but it is important. It is regulatory T cells in respiratory ways, and they play a very important role in restricting all those immunopathological processes in lungs, whether it is respiratory disease or flu or COVID-19. So these cells play a very important role in the majority of respiratory diseases. So they prevent the body from this condition. And another example, this is experimental infecting of volunteers, and there's a very good correlation between the symptoms that were expressed and the number of specific T cells in serum of these volunteers. For flu, we see that elimination of these regulatory memory cells. Okay, if we eliminate them, then there are more lung damaged tissues, and this is in those animals where that did not have regulatory cells, and they have very serious, severe damage in lungs. In such a way, we can say that these infections might be combated by mucosal vaccines, because they form the immune response in the mucosa of respiratory airways, and they contribute to cross protection. So it is protection of some previous variants of the virus, and also it prevents from transmitting this virus from one person to another. Our platform for creating these vaccines on the basis of attenuated flu vector. And we remove NS1 protein and we substitute it with some other protein. It can be mycobacteria or flu or some other proteins, or it can be SARS-CoV-2 proteins. This leads to the following. When this strain gets into the nasal area and gets into epithelium, okay, it expresses this protein of interest, and then all these immune responses are activated that we expect from them. And the vector itself, because this NS protein is removed, it has very good immune activation functions, and the congenital immune system is activated, and lots of other processes. And the design of such structures, we know how this can be modeled. And in fact, we can develop strains with some specific functions. So what are major advantages? Okay, intranasal application. So we also have well-developed systems. So in Russia, in the Soviet Union, flu vaccines were produced for many years. And these schemes can be used for this type of vector vaccines if we use some chicken embryo substrate, or it can be on some cellular lines. An additional effect will be good protection from flu, from that strain on the basis of which this vector was designed. And in terms of the status of development of some candidate vaccines, they are developed for immunotherapy of TB, lung TB, associated with drug-resistant strains. This is a very important area. We work together with medical specialists. Now we identified those proteins that can be and should be used. And this helps to shorten the therapy period, and it becomes more effective. So there are vaccines. We work on the vaccines together with Strategic Planning Center. So it is preclinical trials now, and it is against some acute respiratory disease. And now we are working on COVID-19 as well. We are completing the preclinical trials, and next year we are planning to do the first and second phases of the clinical trials. And another area, it is oncotherapy. It is glioblastomas. And we study the potential of these flu vectors in vitro so far and in animals. Okay, the main results of TB vaccines. This was the first study in humans, first in the world, and it was demonstrated that sublingual and intranasal application gives very good immune response, humoral and local, and these vaccines are safe. And we are planning, we are going to continue these studies. As for RSV, we completed preclinical trials. We see that it is accepted very well. And so there were some failures with aluminum vaccine. So now it is not used, okay, and it has been proven in mice, cotton rats, and calves as well. And we demonstrated activation of all the regulatory and resident cells. So the immuno response is what was expected. And it seems that this vaccine will be most effective for older people. Right, now the latest developments. This is Fluvac preventive vaccine, and we are planning to use 2 doses, and we have 2 strains. One is H1, the other H3, not to have any negative response of antibodies to the first one. And here you see the expression of this NP protein of the flu, and we selected that nucleocapsid protein, and it was demonstrated that this protein is equally effective in terms of the effects that we expected in hamsters, Syrian hamsters, and we used other animals as well. And we are finishing all toxicological studies, and we are planning to begin studying it in humans in 2022. And in conclusion, summing this all up, our brief overview of our mucosal vaccine developments. So there are a lot of advantages, and there are more of them than limiting factors of such vaccines. For flu, it is being resolved and I support what Mr. Loganov said during the plenary session, that it is necessary to use vaccines from different platforms, from different types, and such schemes, such combinations, and these combined efforts will make it possible for us to resolve all the outstanding problems. Thank you very much. And in conclusion, I would like to mention those people who took part in this work. There's various institutions. So Pulmonary Institute from St. Petersburg, and Institute of Medical Primate Studies in Sochi, then a Military Medicine Institute of the Russian Federation. They helped us a lot with experimental models of SARS-CoV-2. And the Institute of Clinical Studies and our colleagues from Kazakhstan. We began the tuberculosis project with them. So partly this was done in Almaty as well. Thank you very much. If there are any questions, I will be happy to answer.
Well, thank you. You were within the time limit. Thank you very much. No other questions? Or we leave them for the discussion time. Okay. All right. Thank you. Then I suggest that we give the floor to Susanna Harid from the Scientific and Clinical Center for Children, comparative efficacy and safety of vaccines against coronavirus infection. Thank you very much. I hope that you can see my screen, and I would like to say that we're looking at the factors that are expected to improve the quality of life and the life expectancy. And the vaccine prevention involves several specialists, several areas of professional work, creators of the vaccines, and we heard their presentations, and practical medical doctors who are carrying out this work. I belong to that particular second group, and for us, it is very important to understand how effective and how safe the vaccines are, in order to to inform our patients. If we take vaccination against coronavirus, in spite of all the new variants, in spite of the strains, in spite of the virus mutation, WHO, when Delta appeared, used to say that in spite of the somewhat lower efficacy of the vaccines, still the vaccines represent the leading instrument to overcome the pandemic. And today, with the emergence of Omicron, this is being repeated. Yes, vaccination is the best measure. So the collective immunity that is discussed so very often is not that clear yet what it's going to be like, particularly since there are very different effects on the virus circulation, even in association with vaccination. But there are some studies that indicate that even the family, most of the members are vaccinated, and the risks for other members of the family are much lower. I'm not going to speak about the vaccines that are being used. You are very well familiar when they produce, the manufacturers are the practical doctors here. But all these vaccines were registered on the principle of the on-face registration. So under these conditions, it's very important to monitor the safety and the efficacy of the vaccines in real practice. So if we take safety, then of course we are very much interested in adverse events, which are very often exaggerated. But here is the real data published for today. The first thing they began to discuss in the context of vaccines on the basis of matrix RNA, anaphylaxis reactions from 1 to 3 for 1 million doses. And the second complication that was also associated with vaccines, that is the myocarditis in young adults. And the incidence was somewhat high in the population than normally, so this made it necessary to discuss the issue of the severity of this status, and say that the risks of the infection are much higher today than the risks associated with the vaccine, which defines the necessity of the vaccine use, the vaccines on the basis of matrix RNA. As for the vector vaccines, the safety issues were also actively discussed as soon as the publications appeared concerning the thrombocytopenia, and again, it was necessary to consider the incidence, the number of those who were infected with coronavirus, and all the data showed the advantage of the vaccination in spite of the certain number of adverse events. Now, if we take the vector vaccine, then quite recently, a lot of attention was paid to the cases of the registration of the syndrome Guillain-Barré. And well, with any vaccines, periodically this registration occurs. Usually it is identical to what happens within the population, so there is no immediate correlation. But the situation requires special attention and observation since the vaccines are quite new. And the data published provide the same conclusions. Yes, this requires observation. So, of course, our patients are always very much interested if we are hiding some events when the vector vaccines are used in our country, for example. Here is the data published in Argentina. For 1.5 million doses of vector vaccines, Sputnik V. There were registered 5 cases acute allergic reaction and 2 cases associated with platelets. And so these were the adverse events registered. And you can see they were possibly associated with vaccination, but this is much, much lower than we can find in the United States and elsewhere. If you take the third dose of the vaccine, how dangerous it is, the booster. Nowadays, the boosters are very often discussed, and what kind of reactions there might be. We can see that different studies show that the third dose, the booster dose, does not give the reactions any different from the first 2 doses. And the booster is being very carefully studied. So it's possible to use the vaccines based on different platforms to reduce the number of reactions and possibly to increase the efficacy. We can see that as for the, if we look at the vector vaccines, and Johnson data is represented here, all these data indicates that the reactions occur in 50–70% of cases. Now, there was a study carried out in San Marino. These were reactions. These were not adverse events, but these were reactions. The reactions might occur with any vaccine, and our vaccine gives the same type of data. The same studies were carried out for different age groups, and they demonstrate exactly what was registered for the during the clinical trials and what we can see when we analyze the vaccination of our colleagues. Yes, there are overall reactions, and about 50% of people do have some reactions, but they're short-term and can be easily recouped. What is the efficacy of the vaccines? A very important aspect which once again supports us in the necessity to use vaccine for different age groups. Comparative study, once again dedicated to the comparison of the efficacy of 5 vaccines to RNA inactivated and 2 vector vaccines, Russian-made, in Hungary, carried out in Hungary with a large population. As a result of this population study, the authors are saying that the vaccine demonstrated much higher efficacy than expected during clinical studies. And if we compare the efficacy of our vaccine with other vaccines from abroad, we can also see that it is very effective preventing death and acute diseases, syndromes. And then a study carried out in St. Petersburg once again proves its efficacy in preventing the disease with lung damage. And the authors are writing the higher is the lung damage, the more effective the vaccine is. So that's the lung damage occurred 78% less often. But not a single vaccine provides 100% effect. Not a single vaccine can be absolutely safe. And if we take the efficacy indicators, we can see that in the United States, the data was published for RNA vaccines, and it says that the breakthrough infections occur in less than 1% of the vaccinated people. The heavy, severe cases and death are usually associated with old age patients. This is one of the reasons for vaccinating and for boostering old age patients to protect them. Any vaccine with any level of efficacy with time loses its efficacy. And there are calculations that indicate that this decrease of efficacy occurs within 1 year by 5 to 7% from year to year when we examine models, of course. And even the most effective vaccines, let's say the vaccine against measles, leads to the lowering down of the immunity by 0.2% every year. So that means revaccination is needed. It's necessary to understand this in order to find the proper arguments to promote your position. As for immunization of different age groups, here we can see the example of Israel, and we can see that for the new strains, the efficacy is lowering down. That means that booster dose is needed, and the efficacy of the third booster dose is important for the same booster and for the different type of the vaccine. And unfortunately, today with Omicron, the situation is going to be the same. Delta shows that the vaccinated can be infected and can spread this virus in the environment, and hence there is a very important thing associated with the extra measures, and the practical medical system should focus on that. Non-specific methods of protection, such as masks and hand washing and so on. One of the studies indicates that in spite of the fact that the vaccines can have different efficacy, our final result will depend on the vaccination coverage and on the use of those extra measures, which, as I said, are very much needed. It's masks, and it reduces the risk. And this also makes it possible to increase the efficacy. Wash your hands. That's another very important thing to mention. A very important factor of the transfer of infection, particularly true for children. And the incidence among children is much higher among those who do not wash hands. So the data we have today for efficiency and for efficacy and for safety make it possible for the practical medical system to be sure of success. Thank you.
Спасибо большое, Сусанна Михайловна, за очень...
Thank you very much for this quite interesting overview and your institute data. Okay, so now we are going to see if there are some questions. So, Susanna Yurina, can you please stay with us? Because there might be some questions. Now, our last report, phenotypes of post-vaccine immunity when using peptide antigens for preventing COVID-19 among people who had coronavirus infection. Dear Alexander Galibovich and dear colleagues, for the second year we have lived in the pandemic, and the statistics that we have 250 million infected and 5 million dead, of course they are enormous. During this time we studied all peptides responsible for such dramatic events, and we know that leading proteins are S and N proteins, and antibodies are targeting them, and antibodies have different levels of virus neutralization functions, and this means protection from the virus. But there are some questions. What about those who had this infection? What happens with the immune system? Is their immunomemory, how long does it stay, how reliable it is, and what are evaluation criteria of this memory? We analyzed 400 people, how long these antibodies stay to S protein and N protein on the right, how long they stay in the body. And we see that throughout a year they remain, but there is a reduction of them, and those And as for the N protein antibodies, they go down sharply. There's no correlation, any correlation of this protein S and protein N results. Now there's an outstanding question whether antibodies do protect from this disease. There's a lot of data that not always antibody response correlates to severity of the disease, to symptomatic or asymptomatic development. And sometimes they say that these criteria are not so reliable if we wanted to learn whether people are protected from the infection. Why is it so? Because in COVID diagnostics, I am an immunology specialist, and among my profession, we all know that apart from the humoral response, there is a cellular response as well, and any antivirus protection is developed at the level of cellular immunity. So we analyzed these 400 people, and we analyzed what happens at the end of the year. Okay, I'm not going into immunological details. So there's changes, significant changes in cellular immune response. And then we asked other questions. How can we increase immunity and improve immunity against these infectious diseases? Can we strengthen this protection through additional vaccination? If it is possible, then how often this should be done? What should be this booster dose? How often it should be used? So these are just some of the questions that we have. We know, and academician Vardalan showed it very well, that the immune system of the population, it is the only barrier for the disease. And this is people who have been inoculated and those who had this infection. And there's a hypothesis there can be some hybrid immunity. For example, if those who had this disease, if they get a booster dose in combination with anti-disease immunity, this might help us strengthen this protection and make it longer. We took part in a project, just a second. We took part in a project of using vaccines at the basis of peptide antigens for prevention COVID-19 among people who had this infection in the past. The purpose was to improve the protective measures and to make this protection longer. Here we speak about some straightforward screening. We selected a certain number of people. We analyzed their status, immune status, both humoral and cellular levels. Then, after we assessed their post-vaccinal immunity, we gave them 1 dose of Epivac Corona, and then 21 days later, we tried to see how the humoral and cellular immunity had changed. The second dose of Epivac Corona was prescribed if there was no response after the first dose or if it was very low. Then we were analyzing during 3 months what changes were observed in the immune system that was changed in such a hybrid format. During the first phase, we had 478 people, and we defined the level of antibodies to N protein. And depending on the expression of these antibodies, we began working with them. We selected 81 people. The criteria of inclusion was some medical records confirming that they had COVID-19 in the past, then low level of specific antibodies to N proteins, less than 700 points. And this work was done together with the Pasteur Institute. And these systems from Pasteur Institute, okay, we identified these G antibodies to N protein. Okay, so here we have these 81 people that met these criteria. We used a wide spectrum of methods, immunoferments. Okay, so specific antibodies can be analyzed, and NST and this flow methods. Okay, so and it was possible to see all the memory cells and regulatory cells, both in I and T systems, and also to assess the congenital immune system. We found out that after the first inoculation of Epivac Corona, 86% of the people, their response was very good. 14% were non-respondent. Okay, there was no response to this Epivac Corona vaccine. So you see all the details below, men, women, different groups. And then we decided to analyze why there was no response in these non-respondents. What mechanisms prevented from this normal response? We analyzed it, and we studied the N protein level, and this red shows non-respondent. Then we analyzed post-vaccine immunity. Again, we were using special systems of the Pasteur Institute. And non-responders didn't have any increase of immunoglobulin G to N protein. Of course, for the first time, we wanted to see what was there in those lymphocytes that prevented them from developing this response. So these people had this infection in the past. Then they got a booster inoculation. Okay, and these RB lymphocytes, okay, this was not homogeneous. Okay, there's B1 and B2 lymphocytes. They have different roles. I'm not going to go into details, but this is what we get. The red line in this diagram, these are non-responders. Okay, so booster vaccine didn't lead to any response. So they had reduced B memory cells. In addition to this, and B regulatory cells also, there were lower of them. So there was some damaged balance of B1 and B2 lymphocytes, and there were more B2 lymphocytes, and they tried to respond to these antigens, but the balance was damaged. Okay, and the analysis of the primary parameters gave an opportunity to analyze how these primary parameters changed after the booster dose. And we saw that B memory cells, their number grew significantly, but at the same time, their amount was not enough for the full antibody response. And the green line shows those people who responded with very high level of specific antibody synthesis. And we see a significant difference here. If we compare this and what cells do not function appropriately, we see that among non-responders, they had lower levels of B regulatory cells, and B memory cells also were quite low, and activation processes do not function appropriately, both early and late response. And taking into account the expression of CD4, okay, we can say that their functions are not enough in forming intercellular cooperation. So in such a way, it can be said, we also analyzed the content of CD3 and T regulatory cells. Again, we didn't see, 21 days later, we didn't see any deviation in the T cellular systems among those whose response was as expected. But those people who didn't respond to it, they had lower levels of such cells. So 11 people who had no response, 11 out of the whole group, they were given the second booster dose. And what is interesting, 21 days later, it is after the second dose, 9 people began developing this response, and 2 people, again, non-responders. And naturally, we analyzed them, and it is immunoglobulin G antibodies. You see 9 people, this is green, the green bar, and 2 people who remained non-responsive. So we carefully analyzed all the changes in the T part of the immune system, and we see that step by step, okay, this was getting closer, okay, and this intercellular cooperation, okay, in comparison with the previous observations. 72 days later, okay, so we continued this follow-up analysis, and we were watching those 9 people who had this response. They have had a stable response, and it was at quite a high level, and non-responders didn't demonstrate any response. And all the changes in B cellular system, and these patients who received a double booster dose, and the conclusion was that there are 2 phenotypes. So when Epivac Corona is used for vaccination, among those who had this infection in the past, there are responders and non-responders. And the basis for this difference is the cells that can be treated as prognostic markers. It is B memory cells, B regulatory cells, and it is a combination of B1 and B2 cells that can vary in different forms. In such a way, if we characterize those statistically significant data that were received, we can make a conclusion that non-responders, people who have poor response to the vaccine, they have a lower balance of B1 and B2. They have fewer B regulatory cells. They have very low activation markers, in particular 21+, and also B memory cells. Again, they are at a lower level. At the same time, for those patients who had normal response, for them, 1 booster dose is enough. That is why throughout 3 months we were observing them, both cellular and humoral immunity, so it was quite stable, and they do not require a double booster, because we know that if we booster up people with antigens, it may have the opposite effect. And in conclusion, I would like to express gratitude to all team members, and without them it would be impossible to reach all the results. And the chief doctor, head doctor, and our rector, who were helping us to work with 400 people and implement this project. Thank you.
Thank you, Lyudmila Petrovna. Your presentations are always most interesting. Any questions? Could I ask a question? Yes, you're welcome. No, practically, how can we tell non-responders from responders? In a practical way, how can we tell responders from non-responders? In a simple way, they do not have antibodies after the booster dose. Well, that means what kind of recommendation do we have here? It's very difficult to understand what is being. Do we suggest that they check after the vaccination, they check the antibodies? We took those 80 people, we examined them, they examined their immune status, and we selected those who already had low count of antibodies, less than 700 of the units of N profits. Then we vaccinated them with 1 dose of Epivac Corona. 86% of them responded with high titer of antibodies. 14 did not, and we called them non-responders. Yes, because they did not respond to Epivac Corona, because we wanted to have the hybrid immunity. And after the post-infection immunity is supplemented by post-vaccinal immunity. 11 people did not show any reaction. So we decided to understand what prevents the response in the immune system. Yes, I understand. In a practical way. So let's say a person who had the infection comes to me. Should I ask them to go through the antibody test? It's too early to recommend anything. We began this work in April this year. So these are the first results obtained by us. And, well, of course, we will keep following the dynamics. And we have a few weeks yet. We have some ideas. The main idea is as follows. Should we vaccinate people once again? If yes, then how many times over? And then over short or long periods of time. So there are a lot of questions yet. It's too early to come up with practical recommendations. Thank you. Any questions? Any other questions? Well, thank you very much. Thank you once again. Now we can start the general discussion. What do you think? Yes, definitely so. I would be grateful to have a discussion, as we have time left for it, while there is a parallel session on the ground floor. When the colleagues join us, then we will be able to start the concluding session. As for the vaccines, I think there is still a lot to be discussed. Now, we had a very interesting discussion during the first part of the day in the morning. I think that we discussed in detail the work on the major vaccines. Sputnik, Logunov spoke about Sputnik Epivac by vector, also discussed in detail. And and Professor Ishmuhamedov also spoke about the advantages of the classical vaccine. So there are still questions, and a lot of those. So right now there was a question asked by Susanna Mikhailovna. Indeed, what should a practical doctor do? When should a medical doctor recommend the second vaccination? I know from my own experience, I was vaccinated Sputnik, I was revaccinated, and I follow the B cell response. And when it reaches, well, consult doctors, of course, and I also have an opinion. So I get revaccinated when it goes down. So following our experience, and not just declaring anything, but I will say that we have a very big institution, 1,400 people. Unfortunately, there were some deaths, and although people are all very well educated and there are academicians among us, but still there were death cases, older age people and quite old people among them, those who were vaccinated and revaccinated. These people, even if they catch COVID, had a mild form of the disease. And so it seems to me that at the everyday level, vaccination resolves a lot of matters. Nonetheless, we do have questions fundamentally important. For example, these three types of vaccines that we have: vector vaccines, peptide vaccines, advanced, of course, with there was the first presentation by Ivan Mikhailovich dedicated to it, and the classical vaccine by Chumakov Institute. Do we need other types of vaccines? In my presentation, I spoke about that. I am not trying to take up too much of your time, but I'm just waiting for your reaction. So I said, and I do know that my opinion is shared by others, that in Russia, vaccine of mRNA platform should be developed because situations might be different. And with this race between the virus mutations and the vaccine response, we need to have another platform. According to the data I could present very briefly, these are preliminary data. Of course, the article is still being prepared for publication. So we looked at the individual cell repertoire of the vaccinated individuals. And so we were able to find the repertoire of neutralizing antibodies with patients who had the full vaccination with Pfizer and full vaccination with Sputnik. Of course, we will test other vaccines as well. In the same way, it's a very expensive examination and survey, so we're looking with just two groups. Both of these vaccines gave a good repertoire response, and we can set out individual neutralizing antibodies. Well, we don't know how active they are going to be against different strains of virus. Now, that is still a question. And no, many clinics in the West and here as well, many of the clinics were on antibiotic resistance. So when a patient comes with a certain infection, and they immediately start the surveillance and control in order to check resistance to antibiotic. Now, this is something that we cannot discuss yet because there are too many spontaneous approaches to that. So among the vaccinal trend, so to speak, if it is not too expensive and if we can use the existing options, then I think that improvement of the peptide vaccines described today, vector vaccines and mRNA vaccines, now all this is worth doing, and the super antigen properties of S protein, that is a very promising trend. Now, my opinion is obvious, I think, and I'm most willing to listen to the opinion of Rospotrebnadzor and our foreign colleagues. You can speak English or Russian, whatever language you choose, or French if you decide. You're most welcome. But the world is going to work for quite a while on the vaccines. The SARS-CoV manifested itself as quite unstable with a T cell response, which is quite weak, and that's my opinion. It's what I can see is like that. I can see there is a weak response here. So that was a sort of an introduction. And now your opinions, the opinions of the participants of the discussion. Welcome, and thank you. Mr. Kruglov from the classical school. Of Sergei Nedospasov. No. This is not an imperative, of course, but if you wish, you're most welcome. No. Yes, I've also been thinking of that. I have some experience of work with Western vaccines, and it's quite extensive experience. And definitely the situation that we have in the West and in Russia, there is a strong lobbying. And of course, we need to have quite a wide range of vaccines in order to respond correctly to the mutations. Because it's obvious that there, with their policy, they have actually failed. And in Europe, as you know, they're lobbying the RNA vaccines, and all the others seem to be pushed aside. And the immune response to mRNA vaccines needs to be studied. On the one hand, it's easy to make them, to produce them, but they, in the past, were never used for people. So the immune response there is different. And so that means we most probably need to combine vaccines. And that's what I can conclude on the basis of work in Russia and outside. I'm talking about Pfizer and Moderna. They're basically the same. Yes, the S protein is just inserted there. Now, for the second time, I would like to repeat, then of course the prospects for RNA vaccines are there. It's very good that they started producing them right away. What other ways there were? Other colleagues were able to play with epitopes, and they managed to carry out their work, and they just worked with S protein. So, but there is. If we variate epitopes, this might work better, probably. But as for neutralizing antibodies, that's very similar indeed, because we are looking at the interface and we induce there, and here they introduce the therapeutic agent. The antibodies demonstrated, the Chinese showed this response. But still there are questions, and indeed the article is very good, but disputable. And in this case, the future is with the cocktail. As for AstraZeneca, AstraZeneca is almost the same as Sputnik in basically. And Johnson & Johnson. What about Johnson & Johnson? No, I know in Germany, for example, the situation, but there was just mRNA vaccines left. No, just those. That is because it was developed by the Turkish German, so to speak. Right, and but it's necessary to diversify, and hybrid immunity is the best immunity. Professor Sizekin spoke about that, and her presentation corresponds with the social situation. Many of those who were immunized with Sputnik for the trip to the United States are carrying out these experiments with hybrid vaccination, and their experience should be properly studied. And this, well, having this microfluid system, I involved some of my staff members. And there's an interesting picture indeed. Those who had the disease and were immunized by Pfizer give a very strong response indeed. I'm not going to give the names to you, but there are some individuals whom you know and whom we studied, and we were able to see an outstanding result with neutralizing activity of the antigens. So this is not the time for vaccines at the moment, but I would like to say once again that we should not forget the low molecular combinations that will support the patients, the metabolism of virus. And so is there anyone else who wants to take part in our discussion and say a few words about the vaccines? Right. There was a very good presentation yesterday, but maybe within Rospotrebnadzor there are certain other approaches you would like to discuss. No, as for that's, yes, Oleg. As for, we do not work on vaccines, but this topic for us is very important. The use of the vaccines, the application of those, and all the matters that are associated with that. And today, not for the first time at all, I'm asking questions. And I would like to ask a question once again. Because you were not among the respondents to my questions yet. So for me, it is obvious that mucosal vaccines are needed. Sputnik is there. It does exist. It's clear. But how effective mucosal vaccine is going to be by itself? Not in the combination with the traditional vaccines, but as an independent substance. That is my first question. And then, what is the niche for such a vaccine? What is its place within the system of vaccination? Because at the time of the epidemic, when half of the population are infected, and infected, that means the epithelial cells of the respiratory tract are suffering, then the application of the mucosal vaccine, or rather its efficacy, from the theoretical standpoint, is doubtful. But in peaceful life, so to speak, this might work differently. Anyway, so that's important to discuss this matter, I think. Right.
Я должен сказать,
Well, Eric Alchamovich, I'm not a practitioner, I'm a chemist and a physicist. But I can say the following. So if we obtain certain knowledge, okay, so we are developing various environments for the Sputnik vaccine, and we work a lot with the Ginsburg group. Of course, without naming any people, okay, and using the term non-responders, okay, several cases, okay, some academicians, they didn't have good response after 2 doses of Sputnik, and after an intranasal vaccine was given to them. Okay, it was an experiment because it was not allowed, so they demonstrated a very good response. Well, and we were discussing these cases, and we were looking at the B cell response and so on. So I know myself experimentally that it worked very well. Their response was very high, okay, and then they traveled abroad and they were among infected people, but they were well protected. But so it works very good for children, for example, yes, socially, yeah, so it will be quite appropriate. But on its own, mucosal vaccine, I don't know, it seems that I agree with you that it won't be so effective on its own. Right now, I'm passing the floor to a friend and a colleague from Mongolia. You wanted to say something. Okay, I'm quite far, so you can remove your mask. Maybe it will be easier for you. And you're going to speak English, right? Okay. Okay, everybody who would like to listen, yeah, they may listen. Okay, yes, thank you very much. The floor is yours. You would like probably to share your experience of vaccination. Okay.
Thank you very much, all of the organizers, and the meeting was very successful, and we have many answers for the many questions. So I think that we should improve the next time what kind of vaccines and working about maybe the booster doses, either heterogeneous and homogeneous, maybe in our cooperation is big in Russia and Mongolia and other countries. So it will be, I think, nice study will be.
So you propose to make some comparative studies, yes?
Yeah.
And may you disclose us what kind of vaccine are you using in Mongolia?
In Mongolia, we use Sinopharm,
Sinopharm, yeah.
Sputnik.
Sputnik also, yeah.
Yeah, and also Sputnik Light. And also
but Sputnik Light, to tell you frankly, is the first component, yeah.
And we also use AstraZeneca and Pfizer.
Pfizer, yeah.
Yeah.
And I think the data which were very nicely proposed, displayed in Hungary, I think two presenters used this data from Hungary, because this country also possesses a wide variety, very good spectrum of the used vaccines. So you showed us in Mongolia that you have also this kind of approach, so you have different vaccines. And do you have some statistics? Do you have some profound studies? What is better, or what is preferable as a type of vaccine? Or it's maybe commercial secret?
No, some groups studied about that, and so around After 2 doses, around 3 months later, the antibody titers declined. So if that time we take booster doses, in that cases,
In case of Pfizer also, you have booster. The same,
all
yeah?
for vaccines, it's same declined after 3 or 4 months. And during that time, when we take the booster doses, in that time, the antibody titers is increased.
Yes, as far as I know, in Europe also, Glugov, they also now insist to use the 3rd and the 4th year shot of Pfizer. I think this is the situation, yeah?
Yeah, so basically the situation is that it doesn't matter what you got, at least in Germany, so you get now Pfizer or Moderna.
No, but they also insist to have the 3rd dose of 4th. Yeah. Yeah,
it's scheduled automatically, basically.
And how long this 4 months or 6 months? 6. 6 months.
Yeah.
Because I think that in France they now propose to make once for 4 months, but in France the anti-vaccine strategy are very how say important. So well, thank you very much for your personal view, and certainly for me, it's it's not the problem is certainly linked with vaccination, but this is a little bit strange that we have a rather high percentage of mortality in our clinics, even very good clinics in Moscow, as I don't know, for example, Sklifosovsky Institute and other. They have very good equipment, very good therapeutic antibodies and so on, but still we have a rather high percent of mortality. Yes, thank you. Maybe
thank you.
Sorry?
Thank you.
Yeah, how is in Mongolia? You have less, the mortality in Mongolia is less from the...
yeah, mortality is less. Fatality case rate is 0.3.
0.3.
Yeah,
okay.
The world is around 2.
Yeah. So thank you very much. So our colleagues from other countries are also very welcomed to...
Alexander, sorry, I have a proposal. I am looking at Mr. Kiyasov. If now we are exchanging opinions, and if you can tell us about Kazakhstan. As far as I know, 4 different vaccines are being used there. Okay, I'm not asking about some comparative study, but maybe empirically, some preliminary level, maybe you can share with us. Is there any assessment, or you are just taking whatever vaccine which is available? So can you please comment on this? Thank you very much, Artyom, for this question. Yes, definitely. Today we are using 5 types of vaccines. The first one that we had, so it was launched on the 1st of February this year. It was Sputnik V. In March, we localized its production in Kazakhstan. It was in Karaganda farm, and the substance, the feedstock, it was provided by Russia. And 80% out of all who has been inoculated, they got Sputnik V. We also had Sinovac, It was manufactured in the United Arab Emirates. The volume was not very high, and currently we are using Chinese, Sinopharm and Sinovac. In addition to this, we are producing our own Kazakh vaccine. It is on the basis of Research Institute of Security and Medical Safety. And we gave choice to our population, so they could be vaccinated with whatever vaccine was available. In November, we began inoculating people with Pfizer and Comirnaty. But this is used only for children from the age of 12 to 17, and also pregnant and nursing mothers. This is a priority. And starting from December, we began booster vaccination, mainly with Kazakh and the Chinese Sinopharm, which is Viracell. Of course, now it is too early to speak about any conclusions. So we wanted to begin this vaccination campaign as soon as possible, and some steps were taken in order to overcome the resistance, because anti-vaxxers movement is quite strong. So we were introducing some compulsory requirements for target groups, and we developed new mechanisms in order to promote the usage of various vaccines. And we were developing some new conditions, and we were using information systems for that. And right now, we reached about 48%. This is the level of vaccination. So this is the whole population. And in the target group, over 70% was vaccinated. And this will be increased. And we are planning, beginning from the previous year, from the next year. So if we have certain agreements, and if we can do it not only through public medicine channels, but in the commercial sector as well, then maybe there will be a greater choice of vaccines. So this work is being done with different manufacturers. I hope it will be successful. And now we see that due to various political aspects as well, it is hard to cross the borders, and some governments prevent travelers from other countries to cross the borders. Or sometimes only people vaccinated with some specific vaccines, they can cross the borders. Sputnik V. So now it is going through the procedure of certification, international certification. But there are certain political decisions, and it seems like it is compulsory now for general population. So we hope next year we are going to overcome lots of these barriers, and we hope that all our citizens will be inoculated. Right. Could you say a couple of words about Kazakh? Yes, a drawing, an analogy. So it is similar to what? Kazakh, it is an inactivated vaccine. Okay, so it's been through all stages of clinical trials, and we submitted documents to the World Health Organization. We expect this vaccine to be acknowledged. Those who were developing it, it is in the team in the Institute of Biological Safety. It is a well-established institution, and they were, developing vaccines in the Soviet times. It is in Jambul region. And on the basis of this institute, additionally, within a very short period of time, we implemented the project of factory construction for manufacturing of vaccine, and also the CL laboratory of class 4 was built as well. And also we are planning to combine a number of research institutes, so they will be all united within one structure, Kaz Biopharma, and they will be working not only on scientific projects, but they will be manufacturing drugs and other biological therapy drugs. Okay, so right, this is an ideological analog with Chumakov. The Chumakov vaccine. Right, okay, we need to finish, because there will be the final concluding remarks. Okay, Kazakhstan, a couple of words. In our country, we use seven types. Okay, Sputnik V and Light, including. It is in Uzbekistan, we are comparing all the vaccines, and AstraZeneca, and Uzbek, and Chinese, and Sputnik V. So we compared them. And the application of these vaccines gives very good immune response and immunoglobulin G and specific response. We also compared it with people who have post-infection immunity, and this post-infection immunity, okay, so the response is not very high, but it has a longer duration in comparison with post-vaccine. Post-vaccine, it is AstraZeneca and Chinese and the one, okay, the higher response, it is almost double as post-infectious, but the duration is shorter, so up to 6 months. We are still studying it, and I can say that after 6 months, we made a conclusion that there should be a booster dose. Also, I would like to say that we wanted to evaluate the heterologous, because we have Moderna and Pfizer vaccination with vaccines on different platforms. It gives a higher immune response. We are not ready to say what combinations exactly, but different platforms ensure a more powerful response in comparison with only one vaccine. We also noticed that heterologous vaccination, it is more reactive. Okay, this hasn't been finished yet. Okay, the preliminary results, they are published in the magazine, okay, and we are going to continue this work, and in about half a year and 15 months, we are going to have some additional quite interesting results.
That was most interesting. There are no representatives of the regulatory agencies, but we must say that when a drug is being examined, for vaccines the rules are somewhat different. Then the joint use of drugs, even certified, is as a problem, because the long-term results. That's a difficult story, because it might all have an impact on the immunity system, sort of disorganizing it. Kyrgyzstan. No, we are also examining the response, and we are using 5 types of vaccines. And the later results show that 91% of the vaccinated individuals have antibodies. We often hear that antibodies after 3-4 months go down with vaccinated, and even with those who had been infected. But if a person is not infected, then the natural immunity goes down. But we wonder if among those who had antibodies, there is a lower level of morbidity. That's what we should think about. Because if we take hepatitis, for example, we know that after a number of years there are practically no antibodies, but there are still some antibodies, and the memory is still there. So that is a key question indeed. So we shouldn't be afraid of the antibodies going down. We really need to check whether these people fall ill or not. With hepatitis, this does not happen at all. The antibodies disappear, but they do not get sick. No, we often say that we do this or that in Russia, in China, in the United States, and what are the viruses doing? The biologists should speak about that as well, because the viruses get adjusted to the bodies. And there is some data already which shows that the severity of the disease is not that high. The symptoms are milder and milder. At the first stage, 40% were without symptoms, and then 60%. And at the Research Institute of Microbiology, it's 70% already asymptomatic, and with children up to 90% asymptomatic. So that means virus by itself is also changing, adjusting, and the virus does not want us to be sick, and does not want us to die. Yes, the virus adjusts to the new environment, to and look at the data for the world. In Mongolia, the death rate is less than 1%. With us, it's quite low. Most probably that's because of the virus that is changing all the time. And as for the mortality rate, just a few things to mention. Let's say in Russia, there is a high death rate, they say, but we should not compare. No, these deaths are not always from COVID, because there is the background level of mortality. And if those individuals suffering from cardiovascular disease would die, no matter what, without COVID, they would die as well. And so this is something to be properly analyzed, and we must have a proper answer to all these questions. And the fake materials, there's a lot of that. We are also saying that 2 or 3 cases associated with vaccine, there are no antibodies, we say. So I think that in the presence of media representatives, we shouldn't discuss that at all, because without getting deep into the issue, they just transmit this information. But statistically, these cases are far from being reliable. So media, unfortunately, are very often misinterpreting the situation here in our country and in the European countries. Very often there is no proper understanding displayed by the correspondents. Thank you very much. I think my mission has been completed, and now we're opening the final session. Rosneft, the academicians are there with us, as they will say a few final words. Thank you. Just one clarification. Your last phrase, I'm quite sure, doesn't have to do with the media representatives who are here with us today. Oh, yes. Yes, this is true. These are always individual cases. This case is far from being representative. And next, it seems to me that here we should not be afraid of regulatory bodies, because we are talking here not about a single-time use of vaccines, but about consistent use of the vaccines with certain intervals. And this is not banned at all. Moreover, it is recommended by Minzdrav for vaccination. It is clearly stated that for revaccination, any citizen has a right to select the vaccine he trusts. Dear colleagues, it seems to me that the organizing committee made a mistake, a slight mistake, planning the conference for 2 days, because our final session and the open discussion shows how active we are. And I think that had it not been necessary to close the conference, this discussion could continue for much longer. So maybe the proposal of your deputy to have a 3-day conference might work for the future. It's sensible and reasonable. But the organizing committee has prepared the resolution draft, which, with your permission, I'm going to read out 2 pages. If there are certain comments, if you like to amend it, then just let us know. So the second International Scientific Conference on the counteractions against COVID-19 and other infections. This was organized by Rosneft and brought together 132 participants with personal involvement and 179 online. There were over 1,000 people took part in the online translation, and there were representatives of these public authorities from the CIS states, from Vietnam, from Guinea, Mongolia, Senegal, and other countries, and the leading researchers and experts of international organizations, including WHO. which is involved in the issues of diagnostics, prevention, and treatment of COVID-19. The welcome address was prepared by the Premier of the Russian Federation, Mr. Mishustin. The new infection is a challenge for the community, and the sanitary and pathological services of the countries and the public health system were under threat, of course. At the same time, that was a stimulus for the development of research work in the field of genetics and different other types of technologies, and humanitarian exchange became very important, and cooperation in the field. The scientific community, including Russian researchers, are studying Omicron and its impact on the development of the processes, on the severity of the disease, and the vaccination. Leading researchers and practitioners who work on these problems made their presentations. Representatives of Ministry for Public Health, of Rospotrebnadzor, Ministry for Defense, and different clinical centers providing support for the patients. The 2 plenary sessions and 7 sessions with 2 roundtable discussions. The main areas of research were discussed, and including the prospects of different new types of preparations, the clinical course of the disease, immunology of the infection, efficacy of vaccines, and antibiotic resistance, and so on. The participants noticed how important the contribution of Russia is into this struggle. And science is a very reliable support for the development of diagnostic means and methodologies and for decision-making process. The rapid algorithm of development and for diagnostic preparations provided the possibility of mass testing. This will be the basis for the reaction to emergencies of sanitary and epidemiological type. And the role of the authorized authorities in the field of sanitation of the Eurasian Union countries and CIS countries, and the work they're carrying out to prevent the transport and the transmission. The work they're doing for the coordination of the work against different infections, including COVID-19. In the Russian Federation, 2 centers for cooperation were established for the work with WHO. This will be a very good support for the sub-regional networks following the principles of medical and sanitation rules. The participants of the conference stated that effective counteractions to new biological threats is possible when there is a coordination of the measures and when the participants of the conference are able to take part in this work together. Health and life of human beings, that is the priority for us when we are trying to combat COVID-19 and its spread. And the events and the measures should be taken on the scientific research and proven data. There must be involved different platforms and sites for the exchange of information for active international cooperation. And it is also very important to continue the improvement of the system of reaction, international reaction to the epidemics with the use of mobile laboratories in compliance with the provisions provided by the sanitation rules, international sanitation rules. And the participants are thanking the Russian Federation for the organization of the international conference dedicated to the counteraction against the new infection and other infectious diseases apart from COVID-19.
Все, спасибо, уважаемые коллеги.
Dear colleagues, okay, we don't have very much time. At the same time, I would like to suggest, okay, we had 7 different sections, 2 round tables, and maybe moderators of those sections, maybe if you wish, you can draw some conclusions. So, is there anyone who would like to say some concluding remarks? Maybe I will begin. My section was on international cooperation. Yes, really, this was most interesting. Okay, we had offline and online speeches. Okay, so one Vietnamese speaker was here with us, the other was online. Okay, then from the Republic of Guinea and also Senegal, the Pasteur Institute was presented by an interesting speaker. So we had a very full program, and what we can say, Russia is working together with neighboring countries and with some countries which are far away. So we work with Asian and East European countries, and it is research work and various preventive medicine areas. So all this has been highly evaluated, and there was great demand for all these projects during these 2 years of the pandemic. And this is what we have been discussing here today. Yeah, so this is important, right? So we now, through combined efforts, we need to combat COVID-19. And there's governmental programs. Okay, the government supports all this, and it ensured our smooth transition to this focal areas of research. And here we see that the laboratories and mobile laboratories were provided, and they are proved to be most effective in some rural, remote areas. Another area of work is a training of medical professionals, especially in the sphere of biosafety. And again, this was something in great demand. It's a very interesting work that is done together with Kazakhstan, Uzbekistan, Tajikistan, Kyrgyzstan, Armenia. There are lots of projects and programs. Experience sharing, for example, in Kazakhstan. So we're sharing information both on clinical diagnostics and the programs. They last for years, and they have been most effective in the area of combating COVID-19. And it's better to work with many countries when we speak about cross-border transmission, and no one can underestimate it. And our colleagues with the Republic of Guinea and Vietnam and Mongolia, okay, it gives its results. So it is remote access and monitoring capabilities. And we speak about new emerging diseases and Southeast Asia. This is the region, and African continent. Okay, this is where new diseases develop. This work is extremely important, and we can share experience and reach excellent results. Further development of this work will be continued, because what is done already in the sphere of fighting COVID-19, I'm sure that this pandemic will be over, but there are other emerging diseases. That is why those monitoring capabilities are so important, because this will stay with us, and in CIS countries we have the same epidemiological environment, and it is extremely important to work with all this. So I would like to stress that all sessions were most useful, and the agenda that we were working on, and here we have representatives of the organizing committee, so they did a lot to invite interesting speakers and to develop these topic-specific sessions. And we see that through this prism of COVID-19, we were looking at the research trends which are being developed in the area of epidemiology, diagnostics, all these new instruments with a wide spectrum of applications. We had discussions. We were speaking about all these relevant things, including treatment and clinical diagnostics. We didn't have it during our previous meetings because eventually all these epidemiological measures, they are not only for preventing transmission of the disease, but also to ensure the best conditions for diagnostics and treatment of people. And of course, specific vaccine preventive measures. Yeah, so all this goes hand in hand. So this conference has been most interesting, important, and it is a significant step forward in our understanding what is happening. So this is what I can conclude. If there are some other comments, go ahead. You are most welcome. Thank you very much, dear colleagues. Madam Viktorovich. Thank you. Yeah, you have been absolutely right. Yeah, these 2 days, this conference in St. Petersburg, for us, for my colleagues, yeah, so we have this great impression because the plenary sessions and sections. Okay, so this is, of course, so relevant. This is also important in fighting the coronavirus. Such conferences provide an opportunity to exchange ideas, to identify future development. And what is most important, we understand that when we are together, we are a force that is moving forward this area, and we can resolve lots of problems we are facing. So I'm most grateful to all the organizers of this event, Rospotrebnadzor, all institutes under Rospotrebnadzor, and administration of St. Petersburg for holding this conference. I hope we are going to continue, and these conferences will be for the benefit of health of the populations of our countries. And like today, we'll have a chance to share opinions and ideas. And my separate thanks for a lot of information. So we got so many materials, research results. All our colleagues got this already, and this will be most helpful in development of epidemiological oversight and combating the pandemic. Thank you very much. Are there any other colleagues who would like to say something? If you allow me, I also would like to say a couple of words on behalf of my colleagues. This is a Kazakhstan delegation. We are so grateful for this opportunity to take part in this conference. And it was important for us to share most important projects and programs with our colleagues. Yesterday, we had very fruitful work. We worked together with chief sanitary doctors within the 7th meeting of senior sanitary and epidemiological officials of the Shanghai Cooperation Organization. And a lot of delegations took part in this meeting offline. It is important for us that during such periods of restrictions, all these meetings during these 2 years, they were online. But of course, face-to-face, it is totally different. And you can express your opinions. You may have a detailed discussion of diverse problems. This is extremely important for us. And we had these opportunities. We worked so well. Thank you very much for this unique volume of information that we were discussing during these 2 days. Okay, there was this big wave of research, and this infection during these 2 years became topic number 1 in the scientific community. And we saw it once again that our Russian colleagues, they are looking at this from different perspectives. Thank you once again. Thank you for a very high level of organization, for your hospitality, and let's hope that such forums and meetings will be held in other places and venues, and we are going to plan it in Kazakhstan, maybe in other countries. Thank you very much. Thank you for your warm words. Are there any other people who would like to say something? Well, then let's draw conclusions.
Вадим Маркович, будете? Да. Пожалуйста, Вадим Маркович Говорун, Россия.
Уважаемые коллеги, во-первых, я благодарен.
Now, I'm most grateful to everyone who took part in the conference. It was intense, and you're right saying that 2 days is not enough to discuss all the issues associated with COVID-19. But allow me to say a few words about something quite different. Now, those 2 years of our observation of the process, and COVID is a phenomenon indeed, and so we were observing as fundamental researchers, clinical medical doctors who work with patients. And so we all started examining the pandemic. And several thousand articles were published. Not all of them, unfortunately, are high-quality articles, but there was so much noise to begin with. There was an explosion of information. And COVID is a great phenomenon, enormous, and the conference has demonstrated it. And time comes when it's necessary to analyze what is happening deeper. And time comes to create a certain picture of what COVID is, because if we divide all this into parts, immunology, clinics, sanitation, surveillance, and all this actually diminishes the efficacy of what civilization should do for the prevention. effective prevention. And I would like to wish to all the participants of this conference and other conferences, because there is quite a number of conferences organized, they are all dedicated to COVID-19, to unite the efforts. We need to work together. We need to have a complete picture, a comprehensive picture of the patient suffering from COVID, about the infectious process, virus migration, virus spread, and different other factors associated with the epidemiology. The sooner we reach this level, the better, and the easier it will be for us to take steps forward. Dear colleagues, since the initiator of this event, Anna Popova, is not with us. She planned to be till the very end of the conference, but there is urgent business and she could not stay till the end. So we were entrusted with the task to conclude the conference. So at the end of the conference, at her request, I would like to say that the conference was most representative. Practically all the departments, the ministries of defense, public health, veterinary service, Russian Academy of Science were represented here. So it was a consortium of Russian entities involved, and everyone had an opportunity to speak up. And I would like to thank all the speakers at the plenary sessions, at the sessions. We know that a lot of specialists took part in the discussion online, both from abroad and from Russia. So I would also like to thank representatives of foreign countries and the CIS and Eurasian Unity and Mongolia and Vietnam and Africa for their active participation. And once again, I would like to emphasize that today, what we are observing, what evokes a lot of attention, is the development of the model of how civilization should react to the new challenges and biological threats represented by the new pathogens. And the pandemics might come again and again, but we acquire new knowledge and we develop new opportunities and possibilities for optimal reaction. Of course, over these 2 years, we have done a lot to understand what's happening. Mistakes were committed. There were excessive measures, which is only natural because we really had to react urgently to this extreme situation. But through a joint effort, we will be able to develop the strategy. In the Russian Federation, it is being formulated. It worked at the earlier stages. It is still being implemented now. And one of the slogans of the sanitary shield of the new program adopted in the Russian Federation is life without lockdowns. And so it's a very short phrase, but it's quite meaningful. So at the end of the conference, I would like to express gratitude once again to all the participants, to all the speakers, to St. Petersburg and the authorities of St. Petersburg, our colleagues from the Pasteur Institute, from the territorial administration of Rospotrebnadzor, who did so much to make our work here comfortable. I would like to thank the organizing committee, which drafted the program and did so much in order to implement it. You all were able to get a unique book, COVID-19. The intermediary results of the enormous work carried out are presented there over the 2 years. And I would like to wish all of you great success. Anna Yurievna Popova asked me to render her best wishes to you. Creative success and health. Along this pathway, in the framework of our struggle against COVID-19 today and tomorrow. Two more words about. So it's for the third time that we are meeting here at this venue. For the time, two sessions were dedicated to the phenological problems. This emphasizes the significance of the type of research and phenological research in combating this infection. Of course, the plenary sessions are needed and sessions are needed, but two roundtable discussions we had at the conference demonstrated that live discussion and live communication, free communication, when just the topics are defined for discussion, and there are no slides and no texts prepared, and the speakers feel free expressing their ideas. So the free discussions are very much needed. Indeed, they are needed for the new scientific ideas to be born. And indeed, as well, all the kind words have been said about the organizing committee and the government of the city, but I think we should also thank the technical committee members who ensured our smooth work, not only during these 2 days, but at the stage when we're getting ready to the conference, during a whole month of preparation. So let's thank them for that. Thank you all once again. We meet again. Thank you.